To light the Bunsen burner, we first check the availability of gas in the gas outlet. Afterwards, connect the burner or the gas inlet to the gas outlet using a rubber tubing. Use a match to ignite the gas while gradually opening the gas control valve. Open the air holes until it exhibits three distinct cones, the non-luminous flame.
Venting gases. When using a fume hood, turn on the hood airflow before beginning the experiment. Never place your face inside of the fume hood. Set the equipment and chemicals at least 6 inches back from the hood door. Do not crowd experimental apparatus when sharing the use of a fume hood.
Clean all glassware with a soap or detergent solution using tap water. Use a laboratory sponge or test tube brush if appropriate. Rinse several times with tap water first and then once or twice with small amounts of distilled water for final rinsing. Invert the clean glassware on a paper towel or rubber mat to dry.
For transferring liquids and solutions, make sure to read the label. Some liquids and solutions have their own unique properties, so you need to be careful. A steering rod can be used for the transfer and only the needed amount of liquid or solution should be transferred.
Handling chemicals. First, read the label on a reagent bottle at least twice before removing any chemicals. Second, do not take out more than what is needed to minimize waste. Do not return excess chemicals to the reagent bottle to avoid contamination. And third, properly dispose the chemicals into the respective waste containers after use for safety and environmental concerns.
Measuring volumes of liquids or solutions. Using a graduated cylinder. A graduated cylinder is typically used for measuring the volume of a liquid when extreme accuracy is not required. When reading the volume of a liquid, the eye should be level with the meniscus. Using a pipette. To draw liquid to the pipette, use a rubber bulb aspirator. Attach the bulb to the pipette, compress the bulb, and insert the jet end into the liquid. Admit the liquid by releasing the bulb slowly. Control the delivery of the liquid with your forefinger, not your thumb.
For decantation, transfer the liquid called supernatant into another container with a clean steering rod. Do this slowly so as not to disturb the solid. For filtration, prepare and cut out a 5x5 filter paper and form it into a cone inside a funnel to be used for filtration. Moisten it with a little water to form a seal. Carefully pour the mixture with a steering rod into the filter paper. The liquid that passes through is called the filtrate, while the solid part that stays in the filter paper is called the precipitate. For evaporation, pour a portion of the mixture in evaporating dish. Place the evaporating dish on a wire gauze supported on an iron ring clamped to an iron stand. Heat the dish over a cool flame until crystals begin to appear. The solid remaining after evaporation is called the residue.
Place the beaker or flask on a wire gauze on an iron ring. Make sure it is placed on the center. Avoid heating the liquid rapidly by placing the flame directly beneath the steering rod, followed by a constant steering. Another option is to add boiling chip to the liquid.
The test tube should not exceed 1/3 full of liquid. Hold the test tube with the test tube holder at an angle of 45 degrees with a cool flame. Move the test tube circularly in and out of the flame, heating from top to bottom. Do not steady the test tube over the flame.
Using a drying oven. When solid chemicals are left exposed to the atmosphere, they often absorb moisture. If an exact mass of a solid chemical is required, the absorbed water must be removed before measuring the mass. The chemical is often placed in an open container in a drying oven set at a temperature well above room temperature for several hours to remove the absorbed water. The container is then removed from the drying oven and placed in a desiccator for cooling to room temperature. When the container containing a solid chemical is cooled, moisture tends to condense on the outer surface, adding to the total mass. To minimize this mass error, substances and mixtures that may tend to be hygroscopic are placed in the desiccator until they have reached ambient temperature.
Glass manipulation, cutting. Place the glass tubing on a flat surface. Make a scratch using a sharp triangular file 30 centimeters from one end of glass tubing. Position the thumbs on the opposite side of the scratch. Break the glass by applying gentle pressure. If this does not work, make a deeper scratch. Lastly, fire polish the edges of the cut glass tubing by rotating it using a non-luminous flame. This is to prevent the sharp edges of the glass from ruining corks and rubber tubing, as well as cutting your fingers. Take a piece of glass tubing about 30 centimeters long and hold it horizontally over the flame. Rotate the tubing on the hot center of the flame. Remove and quickly bend to the desired angle, example 30 degrees or 90 degrees, and hold until it hardens. Reheating and rebending are not recommended to avoid deformation or breakage of the glass.
Collecting data. Record all data entries as they are being collected in data and report sheet. Record it in permanent ink.
Handle the balance with care when it comes to measuring mass. Do not place the chemicals directly on the balance pan. Use a container. Only a professional should try to fix the balance.
All students are required to wear a lab gown during each experiment. Wearing of makeup, shorts, and sandals are not allowed inside the laboratory. Eating, drinking, smoking, and chewing, including gum, are not allowed inside the laboratory. Sitting in the laboratory table is not allowed. No using of cell phones. Horseplay, pranks, and other careless acts are not allowed inside the laboratory. Unauthorized persons are not allowed inside the laboratory. Unauthorized experiments are strictly prohibited. Maintain an orderly and clean laboratory desk and drawer. Discard your waste into the respective waste container. Put all bags and books in designated areas. Do not carry reagent bottles to your desk or lab table. Make sure reagent bottles are in their designated places. All accidents, breakages, and spillages, no matter how minor, must be reported immediately in the instructor.